4.2.1 Law of Dominance. Number 1. Characters are controlled by discrete units called factors. Number 2. Factors occur in pairs. Number 3. In a dissimilar pair of factors, one member of the pair dominates, that is the dominant one, and the other recessive. The law of dominance is used to explain the expression of only one of the parental characters in a monohybrid cross in the F1 and the expression of both in the F2. It also explains the proportion of 3:1 obtained at the F2. 4.2.2 Law of Segregation. This law is based on the fact that the alleles do not show any blending and that both the characters are recovered as such in the F2 generation, though one of these is not seen at the F1 stage. Though the parents contain two alleles during gamete formation, the factors or alleles of a pair segregate from each other such that a gamete receives only one of the two factors. Of course, a homozygous parent produces all gametes that are similar, while a heterozygous one produces two kinds of gametes, each having one allele with equal proportion. 4.2.2.1 Incomplete Dominance. When experiments on peas were repeated using other traits in other plants, it was found that sometimes the F1 had a phenotype that did not resemble either of the two parents and was in between the two. The inheritance of flower color in the dog flower, that is snapdragon or antirrhinum species, is a good example to understand incomplete dominance. In a cross between true breeding red flowered R and true breeding white flowered plant r, the F1 R was pink. Figure 4.6. When the F1 was self-pollinated, the F2 resulted in the following ratio: 1 R, that is red, is to 2 R, which is pink, is to 1 r, which is white. Here the genotype ratios were exactly as we would expect in any Mendelian monohybrid cross, but the phenotype ratios had changed from the 3:1 dominant is to recessive ratio. What happened was that R was not completely dominant over r, and this made it possible to distinguish R as pink from R, which is red, and r, which is white. Explanation of the concept of dominance. What exactly is dominance? Why are some alleles dominant and some recessive? To tackle these questions, we must understand what a gene does. Every gene, as you know by now, contains the information to express a particular trait. In diploid organisms, there are two copies of each gene, that is, as a pair of alleles. Now, these two alleles need not always be identical, as in heterozygote. One of them may be different due to some changes that it has undergone, that is about which you will read further on and in the next chapter, which modifies the information that particular allele contains. Let us take an example of a gene that contains the information for producing an enzyme. Now there are two copies of this gene, the two allelic forms. Let us assume, as is more common, that the normal allele produces the normal enzyme that is needed for the transformation of a substrate S. Theoretically, the modified allele could be responsible for the production of number 1, the normal or less efficient enzyme, or number 2, a non-functional enzyme, or number 3, no enzyme at all. In the first case, the modified allele is equivalent to the unmodified allele, that is, it will produce the same phenotype or trait, that is, result in the formation of substrate S. Such equivalent allele pairs are very common, but if the allele produces a non-functional enzyme or no enzyme, the phenotype may be affected. The phenotype or trait will only be dependent on the functioning of the unmodified allele. The unmodified, that is functioning allele, which represents the original phenotype, is the dominant allele, and the modified allele is generally the recessive allele. Hence, in the example above, the recessive trait is seen due to non-functional enzyme or because no enzyme is produced. 4.2.2.2 Codominance. Till now, we were discussing crosses where the F1 resembled either of the two parents, which was dominant. or was in between incomplete dominance. But in the case of codominance, the F1 generation resembles both parents. A good example is different types of red blood cells that determine ABO blood grouping in human beings. ABO blood groups are controlled by this gene I. The plasma membrane of the red blood cells has sugar polymers that protrude from its surface, and the kind of sugar is controlled by the gene. The gene I has three alleles IA, IB, and small I. The alleles IA and IB produce a slightly different form of the sugar, while allele small I does not produce any sugar. Because humans are diploid organisms, each person possesses any two of the three I gene alleles. IA and IB are completely dominant over small I. In other words, when IA and small I are present, only IA express because small I does not produce any sugar. And when IB and small I are present, IB expresses. But when IA and IB are present together, they both express their own types of sugars. This is because of codominance. Hence, red blood cells have both A and B types of sugars. Since there are three different alleles, there are six different combinations of these three alleles that are possible, and therefore a total six different genotypes of the human ABO blood types. Table 4.2. Do you realize that the example of ABO blood grouping also provides a good example of multiple alleles? Here you can see that there are more than two, that is three alleles governing the same character. Since in an individual only two alleles can be present, multiple alleles can be found only when population studies are made. Occasionally, a single gene product may produce more than one effect. For example, starch synthesis in pea seeds is controlled by one gene. It has two alleles, capital B and small b. Starch is synthesized effectively by capital B homozygotes, and therefore large starch grains are produced. In contrast, small b homozygotes have lesser efficiency in starch synthesis and produce smaller starch grains. After maturation of the seeds, capital B seeds are round and small b seeds are wrinkled. Heterozygotes produce round seeds, and so capital B seems to be the dominant allele, but the starch grains produced are of intermediate size in capital B small b seeds. So if starch grain size is considered as the phenotype, then from this angle the alleles show incomplete dominance. Therefore, dominance is not an autonomous feature of a gene or the product that it has information for. It depends as much on the gene product and the production of a particular phenotype from this product as it does on the particular phenotype that we choose to examine in case more than one phenotype is influenced by the same gene. 4.3 Inheritance of two genes. Mendel also worked with and crossed pea plants that differed in two characters, as is seen in the cross between a pea plant that has seeds with yellow color and round shape and one that had a seeds of green color and wrinkle shape. Figure 4.7. Mendel found that the seeds resulting from the crossing of the parents had yellow colored and round shaped seeds. Here, can you tell which of the characters in the pairs, yellow or green color and round or wrinkled shape, was dominant? Thus, yellow color was dominant over green and round shape dominated over wrinkled. These results were identical to those that he got when he made separate monohybrid crosses between yellow and green seeded plants and between round and wrinkled seeded plants. Let us use the genotypic symbols capital Y for dominant yellow seed color and small y for recessive green seed color, capital R for round shaped seeds and small r for wrinkled seed shape. The genotype of the parents can then be written as capital R, capital Y, and small r, small y, small y. The cross between the two plants can be written down as in figure 4.7, showing the genotypes of the parent plants The gametes capital Y and small y unite on fertilization to produce the F1 hybrid R capital Y small y. When Mendel self-hybridized the F1 plants, he found that 3/4 of the F2 plants had yellow seeds and 1/4 had a green seed. The yellow and green colors segregated in a ratio 3:1. Round and wrinkle seed shape also segregated in a 3:1 ratio, just like in a monohybrid cross. 4.3.1 Law of Independent Assortment. In the dihybrid cross, figure 4.7, the phenotypes round green, and wrinkled green appeared in the ratio 9:3:3:1. Such a ratio was observed for several pairs of characters that Mendel studied. The ratio of 9:3:3:1 can be derived as a combination series of 3 yellow is to 1 green with 3 round is to 1 wrinkled. This derivation can be written as follows: 3 round is to 1 wrinkled into 3 yellow is to 1 green, which is equal to 9 round yellow is to 3 wrinkled yellow is to 3 round green is to 1 wrinkled green. Based upon such observations on dihybrid crosses, that is crosses between plants differing in two traits, Mendel proposed a second set of generalization that he called Mendel's law of independent assortment. The law states that when two pairs of traits are combined in a hybrid, segregation of one pair of characters is independent of the other pairs of characters. The Punnett square can be effectively used to understand the independent segregation of the two pairs of genes during meiosis and the production of eggs and pollen in the F1 capital Y plant. Consider the segregation of one pair of genes capital R and small r. 50% of the gametes have the gene capital R and the other 50% have small r. Now besides each gamete having either capital R or small r, it should also have the allele capital Y or small y. The important thing to remember here is that segregation of 50% capital R and 50% small r is independent from the segregation of 50% capital Y and 50% small y. Therefore, 50% of the small r bearing gametes has capital Y and the other 50% has small y. Similarly, 50% of the capital R bearing gametes has capital Y and the other 50% has small y. Thus, there are 4 genotype of gametes, 4 type of pollen and 4 types of eggs. The 4 types are capital R, capital Y, capital R small r capital Y and small r small y, each with a frequency of 25% or 1/4 of the total gametes produced. When you write down the 4 types of eggs and pollen on the two sides of a Punnett square, it is very easy to derive the composition of the zygote that give rise to the F2 plants. Figure 4.7. Although there are 16 squares, how many different types of genotypes and phenotypes are formed? Note them down in the format given. Can you, using the Punnett square data, work out the genotypic ratio at the F2 stage and fill in the format given? Is the genotypic ratio also 9:3:3:1? 4.3.2 Chromosomal Theory of Inheritance. Mendel published his work on the inheritance of characters in 1865, but for several reasons it remained unrecognized till 1900. Firstly, communication was not easy as it is now in those days, and his work could not be widely publicized. Secondly, his concept of gene or factors in Mendel's word as stable and discrete units that controlled the expression of traits and of the pair of alleles which did not blend with each other was not accepted by his contemporaries as an explanation for the apparently continuous variation seen in nature. Thirdly, Mendel's approach of using mathematics to explain biological phenomena was totally new and unacceptable to many of the biologists of his time. Finally, though Mendel's work suggested that factors or genes were discrete units, he could not provide any physical proof for the existence of factors or say what they were made of. In 1900, Three scientists, Dave Rees, Correns, and von Sesmark, independently rediscovered Mendel's results on the inheritance of characters. Also, by this time, due to an advancement in microscopy that were taking place, scientists were able to carefully observe cell division. This led to the discovery of structures in the nucleus that appeared to double and divide just before each cell division. These were called chromosomes, that is, colored bodies, as they were visualized by staining. By 1902, the chromosomes movement during meiosis had been worked out. Walter Sutton and Theodore Boveri noted that the behavior of chromosomes was parallel to the behavior of genes and used chromosome movement figure 4.8 to explain Mendel's law table 4.3. Recall that you have studied the behavior of chromosome during mitosis, which is equational division, and during meiosis, which is reduction division. The important things to remember are that chromosomes as well as genes occur in pairs. The two alleles of a gene pair are located on homologous sites on homologous chromosomes. Table 4.3, a comparison between the behavior of chromosomes and genes. During anaphase of meiosis 1, the two chromosomes pair align at the metaphase plate independently of each other, figure 4.9. To understand this, compare the chromosome of four different color in the left and right columns. In the left column, possibility number 1, orange and green is segregating together, but in the right hand column, possibility number 2, the orange chromosome is segregating with the red chromosome. Sutton and Boveri argued that the pairing and separation of a pair of chromosomes would lead to the segregation of a pair of factors they carried. Sutton united the knowledge of chromosomal segregation with Mendelian principles and called it the chromosomal theory of inheritance. Following this synthesis of ideas, experimental verification of the chromosomal theory of inheritance by Thomas Hunt Morgan and his colleagues led to discovering the basis for the variation that sexual reproduction produced. Morgan worked with the tiny fruit flies Drosophila melanogaster, figure 4.10, which were found very suitable for such studies. They could be grown on simple synthetic medium in the laboratory. They complete their life cycle in about 2 weeks, and a single mating could produce a large number of progeny flies. Also, there was a clear differentiation of the sexes. The male and female flies are easily distinguishable. Also, it has many types of hereditary variations that can be seen with low power microscopes. 4.3.3, Linkage and Recombination. Morgan carried out several dihybrid crosses in Drosophila to study genes that were sex-linked. The crosses were similar to the dihybrid crosses carried out by Mendel in peas. For example, Morgan hybridized yellow-bodied white-eyed females to brown-bodied red-eyed males and intercrossed their F1 progeny. He observed that the two genes did not segregate independently of each other, and the F2 ratio deviated very significantly from the 9:3:3:1 ratio as expected when the two genes were independent in Mendel's cross. Morgan and his group knew that the genes were located on the X chromosome, section 4.4, and saw quickly that when the two genes in a dihybrid cross were situated on the same chromosome, the proportion of parental gene combinations were much higher than the non-parental type. Morgan attributed this due to the physical association or linkage of the two genes and coined the term linkage to describe this physical association of genes on a chromosome, and then the term recombination to describe the generation of non-parental gene combinations, figure 4.11. Morgan and his group also found that even when genes were grouped on the same chromosome, some genes were very tightly linked, showed very low recombination, figure 4.11 cross A, while others were loosely linked, that is, which showed higher recombination, figure 4.11 cross B. For example, he found that the genes white and yellow were very tightly linked and showed only 1.3% recombination, while white and miniature winged showed 37.2% recombination. His student Alfred Sturtevant used the frequency of recombination between gene pairs on the same chromosome as a measure of the distance between genes and mapped their position on the chromosome. Today, genetic maps are extensively used as a starting point in the sequencing of whole genome, as was done in the case of the Human Genome Sequencing Project described later.